So this is the beginning of chapter 16. It's about the cell cycle and cell reproduction. We will be talking about reproduction on the organism level, but that will come later near the end of the course. So we're going to focus just on the cell cycle itself and how one individual cell makes a copy of itself. Now, the first 2 points I have up there are points that hopefully you remember learning. Even if Science 10 was a long time ago, you might recall doing some sort of activity about surface area and volume ratios. I know in all the classes I teach, I use these pink styrofoam blocks. They're like 100 years old. They have chunks taken out of them. People have written phone numbers on them as jokes. It's very entertaining. But one of the reasons that cells need to reproduce is that there is a size limit to a cell because of this thing called a surface to volume ratio. Basically, the idea is the cell needs to have a certain amount of nutrients enter and waste exit so that it can function properly. And if a cell becomes too large, there is not enough surface to allow entry of enough nutrients to support the huge volume of cytoplasm that's inside the cell. So there is a size limit to cells. What this means is that at a certain point, a cell can no longer get any bigger. It has to make another copy of itself if the organism as a whole wants to grow. The second point is about spontaneous generation, or I really should say the lack thereof. Some of you might recall learning that in the past, people had crazy ideas about science in the past because they didn't have the technology to study things like we do now. But the example I always use for spontaneous generation is that people had this idea, I can take my dirty clothes, it was dirty socks in the example, put it in a pile of hay, and mice will be produced from that. Now, what they didn't realize is that the mice were appearing because bacteria started growing, because there was bacteria on the socks. But spontaneous generation is not possible. All cells are from other cells. And that is the basic principle of cell reproduction that we will be discussing. Every cell that has been created has come from another already existing cell. So a cell just showing up out of nowhere, or an organism just showing up out of nowhere, is not possible. Now, the one exception to this would be the first cell, which we are still not 100% positive how it came about. So I'm not talking about that first cell. I'm talking about all of the other cells that came after that. So all cells come from other cells. So when we talk about cells copying themselves, cells reproducing, there are 2 vocabulary words that you need to know. Mother cell, we would call this the parent cell. This is the cell that starts the cycle. So it's the one that you begin with. It is the cell that is going to go through some steps and reproduce itself, make a copy of itself. The daughter cell, I mean, this would be the baby. I put it in quotations because it's not really a baby. It's just a cell that is produced from the mother cell. But this would be the result of cell division. So anytime a cell reproduces, in other words, a cell divides, the resulting cell or cells are called daughter cells. Now, to talk about what happens when a cell divides and reproduces, a huge part of our discussion has to be about the genetic material of the cell, and that's where this image comes into play. So if you don't have the notes printed out, you can consult page 552 of your textbook, which has a very, very similar looking diagram to this one that's showing different versions of what DNA look like. So if you don't have this printed out, you might want to just open your textbook to page 552 so that you can see the picture that's there. Now, all 5 of these images represent the DNA that would be inside of a cell. The first image, which is also the first image in the picture that's in your textbook, represents what we would call the double helix and and I'll just put DNA here in parentheses. This is a really close-up view of your DNA. The diagram in your textbook gives you a scale of how many nanometers you're actually looking at. But basically, this is the most zoomed in of the pictures, and as we move from left to right, if we were using a microscope, we would be zooming out. So this is the molecular structure of DNA. I'll just point out something here. On this picture, the one in your textbook isn't using the color coding yet because we're going to get into DNA base pairing later. But you'll see that red is always paired with blue and green is always paired with yellow, and that's not an accident. That's very purposeful because when the two sides of DNA meet up, there are particular molecules that have to go together to make your DNA be a double helix. Double means two, so that it has two sides to it. And a helix refers to the fact that it's sort of a spinning circular staircase type of structure. So this is what DNA looks like. The second picture is zoomed out a little bit, so we're not as close. The second picture has what we call histones. Histones are these little proteins that your DNA wraps itself around. So your DNA isn't just sitting around as this loose double helix strand doing nothing. This double helix, this entire structure here, would be represented by the blue line there. So we've zoomed out so you can't see in detail that it's actually two strands. So that's one of the levels of structure of DNA, is that it wraps itself around these histones. As we zoom out a little bit further, we have what we would call chromatin. Chromatin is a long strand of DNA that has wrapped itself around histones. And if you were to look really closely at chromatin, it's not one single line, but it's actually a twirly line wrapped around histones. So that's what chromatin really is your DNA wrapped up around histones in a nice long strand. This is what you see most of the time. So most of the time, your DNA exists this way, as a really long strand of what we would call chromatin. Now, chromatin is hard to see. So if you look at a cell under a microscope and your cell is in a part of the cell cycle where just chromatin exists, it's really hard to see. This image here represents what happens when your DNA makes a copy of itself. So on this picture, you have this little red structure in the center here. That red structure is called a centromere. It's called centromere because it's usually somewhere in the center. Now, is it perfectly in the middle? If you were to measure, not necessarily, but it's somewhere in the middle of the chromosome. You can notice the centromere because usually it looks like the DNA is pinched or sort of strangled in the middle. The word that we use in French to describe what a centromere is literally the stranglehold in the middle of the chromosome, which I think is a really good visual representation of what it is. In this picture, we have represented two sister chromatids. Now that's a vocabulary word that I'll mention today, but that we'll come back to again in our next class. For a cell to divide, it needs to make a copy of its DNA. That's the only way that it can work. Otherwise, it would divide in half, and each cell would only have half of the DNA. So in this picture, if I was to divide it sort of right down the center, on the left is one chromatid, on the right is another chromatid. Chromatids are just versions of chromosomes. So there's a lot of words in this section that sound like the word chromosome. You have chromatin, you have chromatid, we'll have chromosome. There's a ton of words that all start the same way, so you have to be very careful which one you're actually looking at. The last picture here shows a nice chromosome pair. Now, the difference between picture 5 and picture 4 is that in picture 5, we have condensed DNA, and this is actually visible. If we were to look through a microscope at a cell that was in the part of the cell cycle where DNA was condensed, you can see this. Now, this happens much less frequently than, say, picture number 3, where it's chromatin and you can't see it with a microscope very easily. So these are the different sort of levels of structure of your DNA, and throughout this chapter we're going to refer to all of these words as we talk about how a cell reproduces itself. Does anyone have any questions that they want to ask about any of these vocabulary words here? Hopefully you took the time to read section 16.1 and you would have noticed all of these words that I'm writing in here in bold. Perhaps you even looked some of them up. Something that I saw someone doing during scheduled help today was going through the list of bolded words that were in this section and making their own little list of vocabulary, which I think is a really effective strategy if you have trouble remembering what all of the words are and what they mean. Now, this is the second and also the last slide of notes that we're going to look at today, because we're going to talk a bit about chromosomes. This picture, the black picture with all of the chromosomes in it, is found in your textbook on page 552, I want to say, but I'm going to make sure. 553, sorry. There's a very similar looking one in your textbook. So if you don't have this printed out, you can just consult that page in your textbook. What I'd like to do is this. I'm going to pause for a few minutes here, but I'd like to ask you to take a read in your textbook of the section that discusses all of those words. That section starts on page 552 of your textbook and continues on to page 553. And I would like you to, at the very least, write all of these words down. If you feel capable, see if you could put your own description of what each of the words mean. If you need assistance, of course, what it says in the text around the words can be helpful, but there's also a glossary at the back. And then in about 10 to 12 minutes, I will go through and describe or give you some examples of what all of those words refer to. So the first word on our list when we're talking about chromosomes is the term homologous chromosomes. And I'd like to highlight just the beginning of the first word. You have probably seen a word that has that prefix in it at some point before in your life. Perhaps you remember in Biology 20 hearing about homologous structures. Perhaps you remember learning in Science 10 or even grade 9 about a homogeneous mixture. Perhaps you know what homogenized milk is. But the prefix homo at the beginning of a word means same or similar. And so that's the sense that we're using here. Homologous chromosomes have the same gene order. So if I took two chromosomes and I looked at them and they had the same order or the same sequence of genes, then I could say that they are homologous. So for example, if I had two chromosomes and the first gene was for eye color, then hair color, then skin color, in that order, I could say that they are homologous. Notice how I said same gene order, but I didn't say that they were identical to each other, and that's something that I want to point out. Homologous does not mean identical. It means similar to, same order, but they do not have to be identical. And to give you sort of a description of what I mean by that, think about yourselves. You all came from two parents. Each of your parents gave you some of their genetic information in the form of chromosomes. Now, unless your parents are identical twins, which would be actually impossible for them to be your parents then, but unless they were identical twins, then there's no way that the DNA that you got from one parent and the DNA you got from another parent is identical. you would have chromosome number 1 from your mother and chromosome number 1 from your father, but they won't have the exact same information on them. They'll have the same genes in the same order, but which version of the gene did you get won't be the same. So a homologous chromosome has the same gene order, but they are not identical. The second word on the list is autosomes, and the best way to describe what an autosome is to say what it isn't. You have, as a human, 22 pairs of autosomes. An autosome, then, is all of the chromosomes that you have except your sex chromosomes. So if we look at the picture, and there's one just like it in your textbook if you need to refer to it, this is a representation of an entire set of human chromosomes. I just want to point out there are 46 chromosomes. That is the number that a human is supposed to have. That's our chromosome number. You all should have 46 chromosomes. There are a few conditions in which that number could be increased by 1 or decreased by 1, and that's actually the next thing we'll discuss. But normally you have 46 of them. The last pair, so if you have 46, it means you have 23 pairs. The last pair is not a pair of autosomes. The rest of them, from 1 all the way to 22, those are all referred to as autosomes. Your sex chromosomes differ depending if you're male or female. If you are a female, you will have 2 X chromosomes, and these 2 chromosomes will be homologous. If you have an X and a Y, you are a male, and your chromosomes are non-homologous. So you have 22 pairs of autosomes. They are all homologous pairs. The sex chromosomes could be homologous if you're a female, where you have 2 X's. If you are a male, which is what this picture is representing, and you have 1 X and 1 Y chromosome, that would be referred to as non-homologous chromosomes because they do not have the same gene order or the same gene sequence. Now, the next 3 words all refer to something about the information on a chromosome. The first word is gene. If I had to sum it up with one word, I would use the word code. Genes code for something. So a gene is a specific segment of DNA that has a code that makes something in particular. Usually what it's doing is making a protein, and then that protein goes and does something in your body. But if I had to describe what a gene is, it's a section of your DNA that codes for something. Locus is a fancy word of saying location. So each chromosome does not only have 1 gene. And so sometimes we need to discuss what is the location or the locus of a gene on a chromosome. So locus is just us saying, where is that gene? Is it at the end of chromosome number 1? Is it in the middle of chromosome number 2? So it's simply telling us where we would find that gene. And alleles are what we use to refer to the variety of genes that are possible. So if you were to think about eye color is a genetic characteristic, but not everybody has the same color of eyes. Now, you all will have a gene that controls your eye color, but you don't all have the same version of it. There's a variety of different versions of the eye color gene. This variety are called alleles. So when I want to say I have different versions of a gene, I would say different alleles of a gene. So alleles are the variety or the versions of genes that exist. Now the next 3 words all have to do with a number. Now, if you want, I would not be opposed to you utilizing a calculator. You're allowed to on your biology diploma. But I think that you should all be able to do things like multiply or divide by 2. So I'll just let you know, if you really want to, you can make use of one, but I don't think that you need to. This is a part where how many chromosomes there are really makes a difference. Di means 2. So diploid refers to an organism that has 2 of each chromosome. So the part of the word that says ploid is talking about chromosomes and how many of them we have. The way that we would represent diploid would be 2n, sort of a little math notation. The little n would represent the number of chromosomes, and the 2 would be saying, I have 2 of each of them. So a normal human cell, aside from a few exceptions which we'll discuss, would have 2n chromosomes or be referred to as diploid. So this person here, whose chromosomes we're looking at, this is showing you diploid. There are 23 pairs of chromosomes. Now, even though X and Y are not the same chromosome, we always put them together in a pair. So someone who is diploid has pairs of chromosomes. Even though X and Y are not a homologous pair, we would count them as a pair, saying that we have 2 sex chromosomes. Haploid, if you look at the beginning, is talking about half. A haploid organism has 1 of each chromosome. We would refer to that just as N. Now you have some cells, humans have some cells that are haploid. And so the best way to describe where you would find diploid and haploid cells would be to remind you maybe of something that you've learned before that we will come back to. When humans reproduce, we use sexual reproduction. 2 gametes, so an egg and a sperm, each have N chromosomes. So an example of a haploid cell would be an egg cell or a sperm cell. When fertilization happens, and we have what's called a zygote, that's the name of the structure that is made immediately when the sperm fertilizes the egg, it would be called diploid. So if I were to look at the cells that you have in your body, all of your cells should be diploid, except if you are female, your egg cells, and except if you are male, sperm cells. Every other cell is supposed to be diploid. So your skin cells, your bone cells, your muscle cells, those should all be diploid. Poly means many. Now by many, we don't necessarily mean 1,000. Many could mean something like 3 or 4 or 5. But we are saying we have many copies of each chromosome. So I'll just give you an example that I recently heard of. I was reading an article about fish farming. I was actually reading it for my Bio30 class, and then I realized I'm going to show it to you guys in a little bit. But there's an example of a type of fish, I think it was rainbow trout that we were talking about, where they are 3N, triploid. The reason that it occurred is that fertilized or a diploid egg was fertilized. And so normally eggs aren't diploid, normally they're haploid, but in this case it was a diploid egg. It was fertilized by a sperm, which was haploid. You put 2 plus 1 together, you get 3. So these triploid fish were large and they were sterile. They couldn't reproduce. And that's very frequently what happens with polyploid organisms. There's a lot of other examples of seedless fruit that are actually triploid or tetraploid, which means 4. It's a way to get a bigger organism, but maybe not one that can reproduce. Now, you do not need to memorize how many chromosomes anything has except a human. You absolutely must know that a human is supposed to have 46 chromosomes in 23 pairs. 22 pairs are autosomes, and the 23rd pair is the sex chromosome. If I wanted to ask you about a different type of organisms, I would tell you how many chromosomes they are supposed to have. Now, the last little bit here is just a bit of information on how you know if chromosomes are homologous or not. Chromosomes vary based on 3 things. Their length, so are they long or are they short? When we represent a set of chromosomes, we always start with the longest ones. So if you look at this picture or the one in your text, you'll see that chromosome number 1 is always the longest one. The second thing they vary on is the position of the centromere. Now, on little artistic drawings where someone has represented it in sort of a cartoon format, it's easy to see the centromere. On actual micrographs of chromosomes, sometimes it's a little tougher to tell. But for example, right there, where it pinches in on chromosome 7, that's where the centromere is. So remember I said it was like the stranglehold of the chromosome? So where the centromere is something that they vary on. The other thing they vary on is the pattern of color, or if you're looking at it in black and white, the banding pattern that you see on a chromosome. So you would know if two chromosomes were homologous if they were the same length, if their centromere was in the same position, and if they had the same color or banding pattern. When you take an entire set of chromosomes and represent it, that is a karyotype. So this picture is of a human karyotype. So karyotype is just referring to the entire set of chromosomes represented in order. So this brings us to the end of the me talking part.